Welcome to the second part of Virus Simplified. In this particular video, we'll look at the diagnosis, prophylaxis, and a complete overview of viruses causing human infections. In the last video, the part 1, we spoke about the viruses, the general, how they act, what are the types or what are the silent characters of these viruses. Then we spoke about what is the structure of virus, the DNA or RNA, the genetic material. Then we spoke about the capsid, which is the covering of the particular DNA or RNA, the genetic material. After that, we spoke about the envelope, the surrounding safety measures for the virus, how the envelope is produced, that it is taken from the human cell when it comes out of the budding process. Then we spoke about the peplomers which are there on top of the envelope. So some of them are a part of the attachment sites of the virus to this particular human cell. Now there are two different types. The most common ones are what is called as a hemagglutinin and neuraminidase. Then we spoke about the symmetry, what are the different ways in which this capsid and this can be arranged, what are the size of the viruses, right from the smallest parvovirus to biggest one. Then we spoke about different shapes, what's available most, and we also understood that the most common shape is the circular or spherical shape. Then we spoke about the major families of viruses and the major diseases caused by these particular viruses, how they are classified. That's what we spoke about in the classification. This was followed by our replication, what are the main components, attachment, penetration, uncoating, biosynthesis, maturation, and release. So that we have explained in a separate sections. Then we spoke about the genetic modifications, basically the mutations, and more importantly, we spoke about the temperature sensitive mutants. Then we spoke about the interaction between the viral genes, the most common one being reassortment, the genetic recombination, and you have the most important type, which is seen as a reason for most of the infections, which is your viral interference. After this, we spoke about the pathogenesis, how the infection spreads as a local spread, then it goes into a lymph node multiplication and the nearby wherever the draining lymph nodes are. Then we spoke about the primary viremia, which happens when it enters the cells of the reticuloendothelial system. From there, the infection spreads on to the from the reticuloendothelial system cells, it spreads on to the bloodstream again, which is called as a secondary viremia. Most of the time, this is the time when the clinical symptoms are very visibly noticeable. From there, the particular infection reaches the target organs, and we also understood that certain viruses have affinity for certain cells. So those that property is called as a cell tropism, or that is where the virus are very much active, and they are, for example, you will have some viruses attaching only the lymph nodes. Then we found out about the viral pathogenesis happening at different cells. It can be a infection which is an abortive infection, or it can be a steady state infection, and what are the changes that happen inside the host, the cytoplasmic inclusion bodies, which is very important. That is what we saw in the last video. Those who have not seen this video, I would suggest that you please go through that video and you can come back to this one again. So today we'll talk about lab diagnosis. First and foremost, why do you want to diagnose this infection? Obviously, first important thing is to start treatment, and also in cases of blood donation, or when you are taking a blood to the blood bank, it needs to be screened for some blood-borne viruses which can be spread inadvertently. Next, you have the surveillance. You have for any kind of an epidemic or outbreak investigations. There are some conditions where you give a post-exposure prophylaxis, and also to initiate certain public health measures. So what are the most important ones is you can classify them into 4 categories. One is direct demonstration of the virus, 2 is demonstration of viral antigens, third is demonstration of viral antibodies, and fourth is molecular methods. Any virus you talk about the lab diagnosis, will classify them into these 4 categories. So first one, demonstration of virus itself is by electron microscopy, where it can be done using a potassium phosphotungstate and can be scanned under a electron microscope. You can identify the shape, and sometimes you can also directly do the same thing from the specimens as well. And you can also look for the same method using in tissue culture. When you already grown the virus in a tissue culture, you can also use a electron microscope to view the virus. Now, the problem is it is a very expensive equipment, and the procedure itself is expensive, and it needs a good amount of the viruses to be present in that particular sample. A slight modification of that is a immunoelectron microscopy, where you will adding a specific antibodies to make sure that the reactions are much better seen between the antigen, that is the virus, and the antibody. Next one, you have a fluorescent microscopy, where you can the slide is stained with the specific antiviral antibodies with a fluorescent dye, and obviously the fluorescent microscope will be able to pick up the fluorescence, and you will be able to view that. And most commonly this is done in kind of rabies infection or corneal smears, where you take to find out the rabies infection. The next one is a light microscopy, where you do in inclusion bodies. So last video we spoke about inclusion bodies. You can do a histopathological staining or immunoperoxidase staining to look at the viral antigens and the inclusion bodies which are there, and that can be viewed under a light microscope. Here you are not viewing the virus directly, you are only seeing the reaction of that virus to the particular host cell. Then you have viral antigens. Viral antigens, sometimes you will be able to directly detect from the serum. For example, you have a hepatitis HBs, AG, and HBe antigen for hepatitis B infection. You have the NS1 antigen, the earliest marker in dengue virus, P24 for HIV, rotavirus antigen in diarrheic specimens of infection, and the cytomegalovirus specific PP65 antigen can also be viewed. Next part, you have the viral antibodies, and as you all know that as soon as there is an infection, the first antibody to appear is the IgM followed by IgG. So whenever there is a IgM antibody or a four-fold rise in type of IgG, that means it is because of that particular infection only. The IgG alone, if it is present, it can be a chronic infection or because of previous exposure. Not necessary that patient has to have an infection, maybe because of the previous exposure also, which might be a very subclinical infection as well. You have ELISAs, immunochromatography, you have the flow through assays which are routinely used. You have various examples for that. You have the anti-HBC, anti-HBs, HBE, etc., anti-HB, hepatitis C antibodies also. You have antibodies against HIV-1 and HIV-2 antigens as well. Then you have the molecular methods, more specific. They will the results are much quicker. So you have various examples. You have the PCR, reverse transcriptase, multiplex PCR, biofire film array, and real-time PCR. Now the trickiest part is the isolation of the virus, which cannot be grown in your routine culture media like you do in a bacteria. So here it has to be cultivated either in a animal inoculation or egg inoculation or tissue cultures. This is slightly more technically demanding, difficult, and it is not a routine in a virology laboratory. So this requires either for a high-end lab or when you are doing a research kind of a thing, it is very essential that you can do this particular kind of an exercise. So first of all, the animal inoculation. Now you have a lot of restrictions for use of animals in diagnosis. Research, you will be able to use it in a minimal quantity, as less as required. That is for the purpose of studying how the virus cause infection or viral oncogenesis. And sometimes when the viruses are not able to grow well in an artificial medium, you can use this kind of animal inoculation. The first one to come out was an egg inoculation, which was 1931, where they found out that they can inoculate these viruses in different parts of the egg, like yolk sac, amniotic sac, allantoic or chorioallantoic membrane. So every area has a different virus which can grow there and form characteristic lesions. Then came the tissue culture. The first one was they took the organ as such and tried to grow the virus, but unfortunately it has a very limited lifespan. The examples were tracheal ring culture for isolation of coronavirus. Then you have the explant culture where you take the particular cells from the organ, minced tissue, and use it for growing the viruses. For example, was adenoid explants used for adenovirus. Again, that has a very limited growth time, after which the cells become unusable. Then you have the cell lines which are particularly used now. So there also what you do, you take a particular tissue, you will use proteolytic enzymes to separate out the tissue properly, and the cells are washed and counted and suspended in a growth medium which contains all the essential nutrients which allow the particular cells to be alive and active. Then what happens, the cells are adhered to a glass surface on a flask. Here, just like we call media culture plate in a bacteriology, here we call it as a flask. In that, a single layer of cells is layered and it is incubated in the presence of carbon dioxide, and you can allow this virus to grow in that particular cell lines. So there also you have different types. You have the primary cell line which are derived from the normal cells. So what happens, it will allow only around 5 to 10 divisions of these cells, and they'll be very useful for primary isolation. For viruses, for vaccine production is used in this particular type of cell line. So most important ones, what you use more commonly, what you're going to hear is these things, that is your monkey kidney cell lines, the human amnion cell line, chick embryo cell lines. Then you have the diploid cell lines which have a slightly higher rate of divisions, after which the cells will die off. They will also be taken from a normal host itself, and they will have the diploid karyosome. So most common things what you hear is a human fibroblast cell lines for cytomegalovirus. The most common, or the ones which are in current use right now, is the continuous cell lines, because they are taken from the cancerous cells, because they don't die off, or they don't stop multiplying when they come into a contact with another cell, and they are very easy to maintain. So you have a lot of things. You have the HeLa cell line, Hep2 cell line, the McCoy cell line, and the Vero cell line. So this is how the cells look on a culture flask when you have the monolayer of cells there. Now, how do you find out whether the virus has grown or not? So you look for what is called as a cytopathic effect. That is, what are the changes these viruses have caused in these particular cells when they are inoculated. Okay. So for example, you might have something like cytoplasmic vacuolations being formed, or large granular clumps like grape-like clumps being formed in adenovirus, or rapidly the cells become shrunk and degenerated on the cells which will be your, for example, is enteroviruses, or the syncytium formation or multinucleated giant cells can also be seen in this particular viruses like measles, respiratory syncytial virus, and herpes simplex virus. Then you have the shell vial technique, where it involves the centrifugation of the cell culture, and it will enhance the cell contact and viral replication, and you can able to see this particular growth in a direct fluorescence method. Others, you have the detection of viral antigens on the surface of the infected cells, viral genes by PCR, or you can also use an electron microscope to look at the growth of the viruses. Treatment wise, you have various antiviral drugs. Some of them inhibit viral DNA polymerase, like herpes simplex virus, cytomegalovirus, Epstein-Barr virus. You have the next variety which inhibit the mRNA for the cytomegalovirus. Then you have the anti-influenza drugs, which are neuraminidase inhibitors and matrix protein inhibitors like rostamilvan, amantadine, rimantadine, etc. You have a class of drugs which are basically for used in hepatitis infections. That is, again, I provided a list of drugs which can be used there. Then you have the interferons, which are basically the interferons alpha and beta have a viral action. They are produced by cells such as the macrophages, fibroblasts. So these are taken out separately and which can be used for the treatment. So how they act, they don't have a direct action, but they will induce the host cells to produce certain proteins So it can be a part of the innate immunity inducers, and the interferons are available in two formats. One is a human interferons and pegylated interferons. So what is it used? Topically, it can be used in rhinovirus infections, genital warts and keratitis, and systemic use in chronic hepatitis B, C and D infections. So along with this, these can also be used for treatment of some cancers and autoimmune diseases. Prophylaxis, which is the most important part of a viral infection treatment or prevention. So first you had the killed viral vaccines, where this virus is inactivated by heat or any other chemicals, and these are very safe, but they have a very small lifespan, and they might have some adverse effects which can be because of the impurities present there. So that can be prevented by purification of viruses for vaccine protection. Then you had the subunit vaccines, which is a particular component of the virus is taken up, and that using DNA recombinant technology, this particular subunit can be given to the bacteria or yeast chromosome, so that bacteria yeast will produce high quantities of products of this particular subunit, and that can be used as vaccines. Then you have the live vaccines, which is basically you have attenuated, or basically what it means is that you have the virus maintains the immunogenicity, but it cannot cause infection. So they have a stronger and a longer lifespan compared to killed vaccines, and few of them are administered like your oral polio vaccine. The only problem is these vaccines cannot be used in immunodeficiency people or ladies who are already pregnant, because there is a chance of these viruses becoming active and they are less stable than your killed vaccines. Then you have the passive immunization, wherein there is, if the individual is already immunodeficient or there is a case where you need immediate immunization, then you can give human immunoglobulins directly to contract the immediate infection, and then you can think about the active immunization later on. The next part is a combined immunization, where like in the case of rabies, where the patient has a very short clinical infection time, so you can't wait for the body to generate the proper antibodies and counteract the infection. So first of all, you give immunoglobulin on one side and also start off the prophylaxis on the other side, so that the immediate part will be handled by the immunoglobulins, immediate infection will be handled by immunoglobulins, and by the time the body will be able to generate the required antibodies, and it will be able to fight off the further infection later on. So, when you look at the viruses as such, the DNA viruses, the most important ones are the herpes simplex virus. So, it can cause vesicular lesions, encephalitis, some lesions on the skin, genital areas, corneal ulcers. So, for that, what you can look at the Zank smear, or you can isolate the virus in cell lines, and you can also look for the viral antigens, and treatment with the acyclovir. So, varicella zoster virus is two forms. One is a chickenpox and varicella zoster or shingles. And there is cytomegalovirus, which is an infectious mononucleus-like syndrome. That can be, you will be classically seeing the owl's eye appearance of the inclusion bodies. Treatment is ganciclovir and valganciclovir. Then you have the Epstein-Barr virus, which causes infectious mononucleosis, and again you have the treatment for this is basically supportive therapy. You have some Epstein-Barr viruses can also produce some kind of malignancies that can be taken off by acyclovir for hairy cell leukopenia. Then you have the parvoviruses. There is the papillomavirus, one of the most important vaccine preventable cancers, which is caused by the serotype 16 and 18. You have the polyoma viruses, JC virus and BK virus, adenoviruses, which is most common in multiple types of infections, and you have the pox viruses. In this scenario, you also have something called as a bacteriophages. So these are viruses which infect the bacteria, very small ones, which are tadpole shaped, and they'll have a hexagonal head along with the tail, and the tail fibers will go and attach to the bacteria, and the DNA is sent inside using a capsid. So basically you have two cycles. You have the lysogenic cycle and the lytic cycle. Just like we saw in the viral multiplication, the lytic cycle is when the bacteriophage is able to send in the genetic material, the extra genetic material, and it multiplies and destroys the cells and comes out. and whereas the lysogenic cycle is a more stable one where it will combine or stay inside the host cell, and it will keep on releasing the bacteriophages slowly and by little by little. Okay, so again the cycle is the same. You have the maturation and release will release the damage the cell itself. Lysogenic cycle, it will be more of a stable one. So what is the use? You can use it for phage typing. Sometimes it can be used for the therapy also. It can be used as a cloning vector. It's a very important thing in transferring of drug resistance and as a transferring of genes responsible for toxins. Now the RNA viruses, you have the orthomyxovirus, influenza, we all know. Paramyxovirus, very important. You have the mumps, measles, Nipah, and respiratory syncytial viruses. Rubella viruses, very important reason for congenital rubella syndrome. Coronaviruses, we don't have to stress the importance now because you have the SARS-CoV, MERS-CoV, and the COVID-19. The polio viruses, which has been there for a long time, causing aseptic meningitis and paralytic poliomyelitis. The Coxsackie viruses, the rhinoviruses, which is important for the upper respiratory tract infections. You have a group of viruses, the dengue, chikungunya, what is called as the arboviruses group, the Cassino forest disease, Japanese bee encephalitis, yellow fever, and Zika viruses. You have the rabies virus, a bullet-shaped virus as we discussed earlier. The most important, the HIV, the retrovirus type, which is for as a cause as an end-stage AIDS infection. Hepatitis virus A to E, these are all from different families, but the A to E viruses are grouped together because they have a common area where they infect, that is the liver. Apart from this, you have some miscellaneous viruses like hantavirus, arenaviruses, slow viruses, and prions. Prions are very important because they, as such, they don't have any much of a treatment potential right now, and there are some viruses which can cause gastroenteritis also. So this concludes the second part of the virus simplified. I just tried to give a complete overview of viruses, what you need to know. Specific viruses, when we talk about, we'll talk about each and every virus in detail along with the lab diagnosis and treatment. I hope you found this video useful. Kindly like and subscribe to my channel. Share this video as much as possible to your friends. Thank you.